Presentation today is on a different part of the dose response, different model. It's really dealing with something that is important, and it's essentially about the linear no threshold dose response and what it means for you. And actually what it means for you is quite a bit in the world of dentistry, because ultimately you are exposing yourself, co-workers and patients to x-rays, and risks associated with exposure to x-rays is essentially evaluated with a linear dose response model. Many things that are regulated by the FDA and the US EPA are also essentially based upon the LNT model. It has such widespread implications within our society that the spillover effect for the LNT standards in the country probably affects well over $1 trillion in pass-along cost to society. It's actually pretty significant. And it's why we are very much afraid of things at very low doses. It's tied into a belief that that dose responses for carcinogens and mutagens are linear at low doses down to a single molecule or a single ionization. So we're going to take a look at this belief. And how did this belief come to be? And it's, in my opinion, it's a story of mistakes, deceptions, and failed public policy. And I'll tell you my story, and we'll see where it goes. I just want you to make sure that you understand what a linear dose response is, and it's marked in this slide, and it shows that the linear dose response is really showing that the response is proportionate to dose, and that is a straight line linear effect such that you will have a response predicted anyways really down to a single molecule. Now, in the course of science, I believe that sometimes science is best taught as history, and in this case, I think that it really plays out well for the LNT. And it's really some sort of a scientific drama. And there are some key players in this. And it's really important, I think, to know the players and their roles in this. And some are pretty big names. And I'm going to run through these key players now with pictures. This is Hermann Muller. He's the star of the show. He's a Nobel Prize winner. And you can see Hermann Muller getting his Nobel Prize in 1946 from the King of Sweden. Kurt Stern, he was a very close friend of Muller. He was a professor at the University of Rochester and was in charge of the Manhattan Project genetic studies at the University of Rochester. And his work will come to be critical in affecting the reasons why the government, our government, and those around the world adopted LNT. The person in the three-piece suit over here is Ernst Caspary, and he did critical study at the Manhattan Project and ultimately became a professor at Wesleyan University and went back to Rochester. This is Delta Uphoff. Delta was a young graduate student at Rochester, and she tried, as you will see, to replicate some of the Caspary work, and ultimately plays a significant role here. Warren Weaver, he is shown here. He was a director of research for the Rockefeller Foundation for many years, 30 years or so, and he plays a role here because he becomes the chairman of a very critical National Academy of Sciences committee called the Baer Committee, which stands for the Biological Effects of Atomic Radiation. And it is the Baer Committee, under his leadership, that recommended a switch from a linear model to a threshold model. That is why the US government adopted LNT and the world followed this. So Weaver is a critical person in this. Jim Crow is the only person I ever interacted with from this era. Jim died a few years ago at the age of 95, and he's a critical player here. George Beadle was on this National Academy Committee, and Beadle is also a Nobel Prize winner. He won the Nobel Prize in 1958. Became the president of the University of Chicago. William Russell has died about 10 years ago. And Russell was on that committee, that NAS committee, and he was a critical player for that committee and for some subsequent research that I will relate and how it affected LNT. This is Louis Stadler. He became the biggest pain in the rear end in a professional way to Herman Muller. And you can see him here. He dies in 1954 from leukemia, but I'll go over Stadler as well. Barbara McClintock. Anybody ever hear of McClintock? She's a Nobel Prize winner herself. Gets the Nobel Prize in, I think, 1983 when she's 81 years old for this phenomenon which the public called jumping genes. And we will talk about her work. And we have a Russian, Timofey Rozovsky, who plays a significant role in the development of the LNT, and Max Delbruck, another Nobel Prize winner, who worked with Rozovsky in this. And then finally, we have the best friend of Herman Muller. And as it turns out, Herman Muller, when he was going through some serious bad times in 1932, when his world was falling apart, so he attempted to commit suicide by taking basically sleeping pills. And he was at the University of Texas, and somehow didn't get the dose right, thank goodness. And he nearly died, but didn't. But like many people who attempt to commit suicide, he wrote a suicide note. And the person he sent the suicide note to wasn't his wife or wasn't a child, but actually was his best friend, and it was Edgar Altenberg. I actually tried to get the suicide note. It's preserved in his in Muller's papers, but Muller's surviving daughter won't let his suicide note be seen by others. It's only been seen by one person, I think, outside the family. So that's the list of characters, and they're going to come up in this story. And now I'm trying to show you the time framework. When do you think that the concept of LNT came to be? When was the first articulation of this in the literature? Well, actually, as it turns out, the big questions in biology in the first half of the 20th century really were focused on trying to find a mechanism of evolution. LNT, which is going to come out of this work, was not really on the radar screen for risk assessment. It was really it was going to emerge out of other research, actually, tied into mutation. Because what the big race was for from about 1910 to when Muller found it was they wanted a mechanism of evolution, and the mechanism had to be the induction of gene mutation. But people could not induce gene mutation. You would think it would be the easiest thing possible. But from 1910 to 1926, 27, a lot of people tried, and a lot of people could not do it. They had different models. They had plants. They had various types of animal models. They threw everything at them, you might say, except the kitchen sink, different types of radiation, various types of harmful chemicals, extreme heat, other kinds of alterations, and no one could induce a reproducible gene mutation really over a long period of time. And Muller was really focused on this. But in 1926, Muller was able. Now there was a race, as you can imagine, to see who could get this discovery, because ultimately it would yield great fame and fortune, big prizes, and Muller knew this, and he was going for it. So in 1926, Muller actually, according to his notebooks, he actually had demonstrated in his laboratory the induction of mutations in Drosophila. And he had several more experiments to go. And by the way, I have obtained an entire copy of his Nobel Prize notebook. I have all his data, all his this and that, all little squirrely pictures of different things while he was in the lab waiting for things. It is interesting. And so Muller didn't know this, but Stadler, whose picture I've showed to you, was 3 months behind Muller, trying to get the Nobel Prize and trying to be the first to demonstrate mutation. Well, as it turns out, Muller had 2 of his 3 experiments done by late spring, early summer of 1927. And so Muller worked a deal out with the editor of Science to publish a paper on his discovery and to discuss it. He didn't, and this is this major paper to show that radiation could cause mutation. He actually published a paper, didn't show any data, and just discussed the data. It's a very interesting situation. And there was never been an explanation offered by Muller, by Science, by anyone as to how the editor of Science actually permitted, on the most significant topic of the day, publication of a paper discussing the findings without showing the data. And it led people to speculate whether Muller had the data, what was going on with this. He eventually shows up at a Fifth International Genetics Congress in September of 1927, 4 months after the publication of his paper in July of that year, and he presents his data. He has the goods, and he presents the data, and he became instantaneously a major player in the world. He had the goods, and so this is where it all starts at that point. Now Muller had Muller's work was not strong on the dose response, and he could not demonstrate a linear or any type of true dose response relationship. So we got a couple of students to follow up, and they looked at very high doses like he did, and but this time they had about 4 doses, and they demonstrated a linear dose response at very high doses. And in 1930, Muller introduces for the first time in the literature, the concept of a linear dose response, but he doesn't call it a linear dose response. He calls it a proportionality rule. And he said, so basically, LNT, linear dose response, is first going to be known by that term. Well, as it turns out, a lot of criticism came to the surface about 1930-31 with respect to Muller's work, and it was coming out of the work of Barbara McClintock, because when Muller did his work, Muller could not do good cytogenetics. The technology was not good for Drosophila. But McClintock in 1929 developed this technology for corn. And essentially, her capacity for resolution of chromosome structure was markedly improved. And so she went to work with Stadler in 1931 at the University of Missouri, and Stadler had shown mutations 3 months after Muller did, and Muller just beat him out on the time. But Stadler was working with radiation and mutations in 1931 in Missouri, and he asked McClintock to look at his corn cytogenetically. She looked at his corn, and she concluded that what you think are gene mutations are simply chromosomal inheritance that came about as a result of big holes knocked in the chromosomes by high doses of radiation, and then you had rejoining of these chromosomes, and you saw altered traits, but it wasn't really gene mutation. It was chromosomal rearrangement and reconnecting. It wasn't subtle changes that you might expect in evolutionary biology. And she said, you didn't induce gene mutation. And he resisted her, and he fought her, and he eventually was convinced that she was right and that his own interpretation of his findings were wrong. This led him to come to doubt Muller's own interpretation, that maybe Muller hadn't induced gene mutation. Maybe he likewise had poked massive holes in these chromosomes, and everybody was wrong in their interpretation. And so what happened was, from that moment on until Stadler died, Muller and Stadler had a major scientific chess match against each other, in which one would do a study, the other would do a study. They would compare and contrast and duke it out, actually. And essentially, in this battle of what I call radiation geneticist titans, very smart people who thought very differently and trying to figure out what the answer was, Stadler's experiments were designed almost always slightly better, more insightfully, and he, after a whole series of experiments back and forth, had basically backed Muller into a corner where Stadler had come up with a series of alternative interpretations that could explain Muller's findings. And Muller could not challenge legitimately the scientific position of Stadler on this. But Stadler couldn't quite knock him out. Stadler really had come up with a series of very credible alternative explanations which basically challenged Muller's interpretation. But Muller was losing the debate. And then in 1938-39, Muller, smart guy that he was, he came up with a new way to evaluate mutation and dose response. And he tried it because it was no value to come back and do the same what he was doing, because he was losing to Stadler. And so he came up with a new idea, and he wanted to check whether, and you might think of this, if you gave a high dose of radiation and you looked for mutation, you get an answer. But if you took the same dose and titrated it out over many small doses, if you would the damage be equivalent between that chronic study and the acute study if the total amount was the same? Muller was of the opinion that the total damage would be the same, because he thought that the damage was cumulative, wasn't repairable, not reversible, and if you translated that out, it would come up with a linear dose response. And a graduate student of his at the University of Edinburgh did a study that tested that hypothesis. And in his dissertation, he found data to support Muller's idea. And so this was really revived and breathed life into Muller's position, and essentially kept the battle going between him and Stadler. Now, soon thereafter, World War II started. And Muller came back to the United States in 1940. And he actually came back to my town, where I live today, in Amherst, Massachusetts. And he became a professor at Amherst College and was there for 5 years. A number of professors had been drafted into World War II, and he was a little too old, and he filled in a slot there. And so during this time, as you can see up on the slide here, we have in 1943, we have basically the Manhattan Project started in earnest at the University of Rochester, where individuals were trying to figure out radiation and mutation. And there were two large funded areas. One was on mammalian models with mice, and one was in Drosophila with fruit flies. Well, as it turns out, the radiation study with mice never developed any publishable data during the time of World War II and years afterward, and it was led by this gentleman, Donald Charles, who was known to be very particular. He would never publish anything until it was absolutely right, and nothing was ever absolutely right. And he was on this study for like 12 or 14 years, and he never published a paper, even a decade after World War II was over, and frustrated everybody. And then in 1954, Charles resigned from the University of Rochester, and the next year he committed suicide in a hotel room in downtown Manhattan. And nothing really came out of that. There was over 400,000 mice in that study, and they got nothing. And so that meant that what happened was that everything was going to rely upon the Drosophila study. And the Drosophila study was actually going to be a big replication of that Muller thing done a few years earlier, because the Muller study had a number of weaknesses in it, and it really had to be strengthened. And so it was made of two parts: the acute study, and the acute study, all the dose was given essentially at one time for over a very short period of time, over a number of doses. And they got a linear dose response there, just like they thought they would have. And it was done by a very good worker, Warren Spencer. And then Kasperi comes on the scene. He was going to do the same dose, but titrate that out. And he does. And he walks in August of 1946 to his boss, who is Kurt Stern. And he tells Kurt Stern that, guess what? When I did this out in the chronic study, I could not support the LNT. I found a threshold. It's a very significant point, because what really counted was the chronic study, because that's how people are mostly going to be exposed chronically. And so Stern looked at him, and Stern would not accept his data. This is when life becomes interesting now. Stern would not accept his data. He said, your data can't be right. And he said, the reason why I'm not going to accept your data is your control group must be reading high. It just has to be reading high. And if it's reading high, you'll see a threshold and you won't see linearity. I'm not going to accept your data. And I have documentation for these. I have their letter correspondences for and memos and back and forth. I have everything documented. I bought all their papers from their respective universities where their papers are preserved. And so what happens is that Kasperi, even though he was, Stern basically came to that conclusion, Kasperi dug in, and he went into the literature, and he came back with a number of citations, showed him that actually his control group was behaving the way it should be, and that he was incorrect. And he had the goods to support him. And to his credit, Kurt Stern backed down. Now, what happened next becomes really interesting in this story. They wrote a manuscript that would be submitted for publication, and in that manuscript, you have to understand that Muller is a consultant to this study. Basically, Stern is the principal investigator. Stern and Kasperi write the draft manuscript, and in the discussion of it, they spent the whole discussion saying, these are our results. We don't want showing a threshold. We do not want the scientific community to accept our findings until we can figure out why our data differed from the results of Spencer with the acute. And basically, it was almost impossible. It was like a straw man that was set up. It was set up because there were like 25 methodological differerences between these two papers, and it was impossible to really figure out why they could have differed. And so they made it set up so that it was a very weird sort of a thing that you would actually write a paper and ask the people with the strongest study to date, a much better methodological study than the Spencer one, because it was done a year later and they had improved their temperature control, lots of other things to it, to say don't accept our data, that we don't, not that we didn't believe it, but we can't explain these differences. And so you could accept the first study, but you can't accept this. And I think they had that because they really still wanted to accept this LNT interpretation. So what they did was they basically, I have correspondence, because I knew that when Muller got his Nobel Prize and he gave his talk in Stockholm on December 12, 1946, Muller stood up in front of the Nobel Prize audience and he said to them, there is no possible way that a threshold model is credible. You can no longer continue to follow this. You have to switch to a linear model, and that's it. He was very, very strict and firm in this conclusion. And I had read his Nobel Prize speech a number of times, and I said, well, gee, he was a consultant to this study. They had just shown in the strongest study to date a threshold discovery by Kasperi. I said, if he was aware of these data, how could he go to the Nobel Prize audience and say there's no possible way that a threshold could occur? How could he do this? I said, well, maybe he never saw the data. Consultants aren't shown everything. So I went in further into their correspondence, and lo and behold, I found that on November 6th, 1946, Stern sent a letter to Muller, and he included in it the draft manuscript by Kasperi, and had all the data and all the interpretations. Muller wrote back to him with a letter on November 12, 1946, saying, I received the manuscript in the research of Kasperi. I've seen that this is challenging the linear dose response. I don't have any reason to doubt the credibility of Kasperi. He's a good investigator. I don't really believe that this could be possible. What we have to do is you have to get some more grant money so that you can test this hypothesis and see if it's real. And so he said what I think a scientist would say. But when he actually went to his Nobel Prize speech, he didn't share that information with the audience. He gave them his own conclusion that there was no possibility that a threshold could exist. And yet he was actually stating in his letter to Stern that, well, this is a challenge to LNT. No, you have to get money to test this. It would take a whole year to test it once you got the money. And so he was, in my opinion, he was actually being deceptive to his Nobel Prize audience. He could have said, I think most people would have said, I'm not sure what the nature of the dose response is. I tend to support a linear dose response, but there's debate about this. It needs to be evaluated further. That to me would have been the easy, responsible thing to do, but not Mueller. In his moment of glory, he goes to this audience and he puts the weight of a Nobel Prize behind him and says, there's no possibility that a threshold could exist. You have to accept linearity. Now, and it's interesting but I would have to say that Stern did get that money and did get made efforts to replicate the findings. And that's when Delta Uphoff comes into the story. She goes in and she tries to replicate the experiment. And what she finds, this is interesting, she finds that her control group is reading very, very low, aberrantly low. So low that she and her boss, Kurt Stern, wrote in their paper to the Atomic Energy Commission, and it was basically classified, nobody could read it, that they had a problem with their control group, that because it was so low and so aberrant, that their data were uninterpretable. And so she then did a second experiment, and the control group was also extremely low and had the same conclusion. She then does a third experiment, and the control group came out normal, but the response was aberrantly high, like about 4 times higher than a linear dose response would actually show. So she came through these 3 experiments, and basically it appeared that nothing was viable, was going to come out of this Manhattan project that was going on. So what does Stern do? This is when it becomes even more interesting. Stern decides to publish the 5 experiments, the Spencer, the Kasperi, the 3 Delta Uphoff experiments, in a summary paper, technical note in the journal Science, and he does. Now what happens is that he basically took these Delta Uphoff studies that were uninterpretable, and now he's claiming that they're interpretable and that they make sense. And he took the Kasperi study that he initially challenged because of the control group being aberrantly high, and then he backed off on that. He's now discounting the value of that study. And so by then combining with the Spencer study and the 3 now credible Delta Uphoff papers, he makes his claim for a linear dose response. It was only a 1-page note. They didn't show any methods, materials, and so forth. And they claimed that they would provide the scientific community with a detailed paper with all the goodies, the methodological goodies, that all could see what they did. And they never published that follow-up paper. It's very, very fascinating. This becomes a very significant paper in what's going to be the decision to go linear. It's in Science. It's got a lot of pizzazz behind it. Now, over the next several years, if you follow Muller's publication, Muller goes, now I have many papers, letters between Muller and Stern, and they were they were just trying to figure out what was the control group mutation rate. And in this, Muller clearly, unequivocally states that Uphoff's controls were aberrantly low, and that he totally supported the Kasperi interpretation. So he was on board in writing, and I have like 5 or 6 memos to this effect from Muller to Stern, back and forth, back and forth, back and forth, back and forth. But 2 years later, 3 years later, in the literature, Muller goes in and he tries to, and he does. He basically claims that Kasperi's data cannot be used because the control group is aberrantly high, and the Delta Uphoff's data was supported a linear dose response. And I believe he's totally fabricating, totally lying in the literature. He's contradicting his own data. He's contradicting many memos back and forth. And I believe that there's something very fishy going on here over these data and disputes, and it was never brought out in the literature, ultimately, until I got into it. Now, the next thing that happened in this dispute is that there's above ground testing in the 1950s with the Soviet Union, the US, and some other countries, and there are concerns over public health and radiation. So the Rockefeller Foundation gives a big grant to the National Academy of Sciences to have a committee together to evaluate risks from radiation, and they have 6 different committees. But one of those committees, for the very first time, is specifically on genetics and gene damage. Before, genetic was always linked, lumped with medicine. And this was a strategic decision that the Rockefeller and the National Academy put together. You have to understand that the president of the Rockefeller Institute for Medicine and the president for the National Academy was the same person, Detlev Bronk. This is a very interesting conflict of interest in many ways between a funding agency and the donor. It's the same person, and so he was funding himself essentially, and then what they, I believe, also did was they selected people to be on this committee that had very strong opinions as to the dose response, and they were all supporting a linear dose response relationship. So I knew that the answer, the question going in was, what were they going to adopt, a linear dose response or a threshold dose response? But I knew the answer coming out was they went linear. So I wanted to see the debate between these people at the meeting. So I got the transcripts of their hearings, their meetings, and there was no debate whatsoever. The very first day that the group met, Muller's colleague from the University of Indiana got up and read the equivalent of the Apostles' Creed, except it was the creed of the radiation geneticist, and it was all effects were cumulative, they were irreparable, damage wasn't reversible, and the dose response was linear. They had their answer at the very first morning of the very first meeting. Yet they were going to have 4 or 5 meetings because that's what the budget allowed them to do. So not having anything to do, but they had to make up time. This becomes really significant. What happens next is that Weaver, the chair, he asks the geneticist on the committee, of which there were 12, he asked them to go back, take a month, and assume that all the humans living in the U.S. at the time, adults, were exposed, their gonads were exposed to a certain level of radiation, gave them the level, and they wanted to predict the number of adverse effects, genetic adverse effects, in the next generation and for 10 generations total. And they had to assume that the dose response was a linear dose response. He said, well, this will be good because some of you are bacterial geneticists, and some are population geneticists, and some are Drosophila geneticists, and some are whole ranges of different geneticists. And if you all kind of converge with similar numbers at the end of this, then it'll really add confidence to what any conclusions and recommendations we might have. So they all went back, and 3 of the 12 decided that they couldn't do it because it was too speculative, and they decided not to offer any write-ups. But 9 of them did, and I got copies of all the 9 write-ups. Now, I have to tell you that, and I got all their correspondence. Now, the interesting thing is that, so I knew that there were 9, I was familiar with them all, and they published, in June of '56, they published their results, their conclusions and recommendations that they wanted the country to go linear for radiation-induced damage. And they cited in the science paper, they said, and we asked our geneticists to estimate the damage from such and such radiation to the gonads for the next generation and so forth. And 6 of the 12 took up the challenge. And I said, wait a minute, 6 of the 12. I said, I have 9 of these in my hand, in my office. So I went back and in my file, I dug out the 9. And then I go back to the paper and it said, 6 of the 12 accepted this challenge. So So I have a discrepancy here in the National Academy Science paper with their recommendation. So then I go back and I go through all their correspondence and I try to figure out which ones that they eliminate. And they eliminated, I found out, determined from all the correspondence, they eliminated the 3 lowest estimates. And by eliminating those, it profoundly reduced the variability in the estimates of the remaining people. They had correspondence, in the correspondence that came in, Jim Crow, who was collating everything, wrote to Weaver and said, we have a problem. Our problem is that our people are totally at odds with each other. There's so much variability, there's so much disagreement amongst ourselves that if people saw our disagreement, they would never accept any recommendations that we would offer. And so he said, what are we going to do? And so what Crow did, I believe, I have no evidence that he didn't do it other than on his own. He eliminated the 3 lowest. Okay, and we're left with 6. Now, then in that same science paper, if you look at the variability, it had a range of 750-fold variability. But when they wrote up in the science paper, they said in 6 of the 12, and it was and their range was plus or minus 10, so they had 100-fold variability. But the actual variability was out to 750 for the 6, and if you added all the others, it was profoundly wider. And so what they did when they came out with their recommendations, they got front page in the New York Times, the Washington Post, they got big story in U.S. News and World Report. They were all over the place, all over the world, and they were the experts. Now, and it was within 2 years, major groups across the world had essentially adopted low-dose linearity, and it applied it to cancer because they generalized it from reproductive cell, which is what Stern was studying, and then applied it to somatic cells. And all within a flash, the thinking was transformed from a threshold to a linear dose response based upon the recommendations of this Bayer committee, and they were really relying upon the Manhattan Project that Stern had done, which essentially had all the problems and deceptions that I told you. Now, all this is unknown, and the country has gone forth, and in essence, the U.S. EPA and other organizations, they reached back into the Bayer Committee, and they based their recommendations for chemicals and cancer for linearity on the recommendations of the National Academy of Sciences. Now, an important intermediary point here to share with you is that in 1958, in December of 1958, Bill Russell, who was on that committee, working with mice, and he was working not with mature sperm, but he was working with spermatogonia and oocytes. He found the existence of essentially a dose rate phenomenon, and that meant that when you gave the stuff all at one time, the radiation, and you titrated it out, he found that in fact it made a huge difference, totally different than what was reported in Muller's earlier work. That in fact when you had with the female oocytes, even when you gave them 27,000 times above background, if you gave them at that rate, the animals never showed any uptick in genetic damage. There was some repair process going on. And so it was really a threshold that he showed. The males showed 70% reduction in damage, and they didn't go back to a threshold, but they showed a repair process. It was later shown to be DNA repair a few years later. Now, the interesting thing is that by that time, linearity had become established, and in 1970 to 72, a new NAS committee was put together called the Beer 1 Committee, the Biological Effects of Ionizing Radiation, and they re-looked at this earlier 1956 thing, and they realized that life wasn't good if they wanted to base human standards on a fruit fly. And so they wanted to base it on a mammalian model, and Russell's mice were the best thing to do because he had data on over 2 million mice in a big Department of Energy study. But they also acknowledged that the Bear 1 committee made a mistake when they said that there was no such thing as a dose rate phenomenon, now that Russell had proven that there was such thing as DNA repair, and dose rate did exist. However, they stayed with the linear dose response because even though the female showed a threshold, the male hadn't quite gone down to it. And that is ultimately why we have today a linear dose response relationship for chemicals and radiation. It all goes back to that. Now, an interesting thing, and that is that Bill Russell had one PhD graduate student, and this graduate student stayed a whole career at Oak Ridge. His name is Paul B. Selby. Now, Selby was very loyal to Bill Russell, and Bill Russell's wife was also a prominent geneticist. But in 1994, Selby had to go back into their data set. And he found some irregularities in the historical data set that Russell used that was now, that the government was basing our regulation on. And he found that they made in his opinion, a profound mistake in the calculation of genetic damage in the control groups. And so he quietly went through and documented his records and came up with about a 1-inch thick report, because this was undercutting an entire 40-year DOE study of mutations, the thing that our regulations were to be based on. And it was really with his advisor and people that was almost like family to him. So he went to the top administrator at DOE, didn't want to go to Oak Ridge, and he showed his information. This has never really been written up in the literature. I've had about 15 hours of interviews with Paul Selby and others and so forth on this, and so I believe I have my information okay. And well, as it turns out, the Department of Energy believed that he had standing. And they decided to bring in 4 people of some note outside of Oak Ridge to see who was right, Selby or the Russells. And so after duking it out in their together, and I have all their write-ups and how they contested each other, it was decided that in fact, see, Selby was not only were they wrong, Selby was claiming, but Selby was claiming that the Russells actually did this intentionally. And so he was bringing up, scientific misconduct. It was a pretty ugly scene. So the group of 4 did not want to get into that scene. But what they decided was that Selby was right, that there was a discrepancy in the control group data. And they required the Russells and Selby to publish their own independent corrections in the scientific literature. And so within 2 years afterwards, they got their papers out. And believe it or not, the Russells acknowledged that their error was by 120%. They were reading low by 120%, high by 120%. And Selby claimed that it was even higher. And so this was written up in the literature, and it was written in such a sanitized way that it was very technical. It was hard for actually one to really decipher the emotion that was behind it all. But over time, the scientific community has strongly favored the Selby interpretation based upon my reading of the 20 or so 30 papers that have cited these things. Well, the interesting thing is that 2 years ago, a year and a half ago, I went back in and I decided to say, well, what would have happened if Selby's correction had been made in 1972? What would that have done to the risk assessment process? And so what if you made the correction, the Russell correction, it would have resulted in that male being a threshold, just like the female was. And the female would have shown itself to be hormetic. Now, this is if you use Selby's work. Now, the interesting thing here is that what this is saying is that our regulations today, based upon Beer 1, based upon the Oak Ridge Russell report, actually did not show support for a linear dose response. Actually showed report for a threshold or a hormetic dose response. And so all this material has come out within the last 3 or 4 years with publications essentially by me. And And the other interesting things that have come out here as well, and that is modern technology has been able to go back in with gene sequencing and other techniques, and they have actually gone back in to the doses that Muller used in his Nobel Prize research, and actually have shown that he did not induce gene mutation. He actually induced profound chromosomal rearrangements, and that he got the Nobel Prize for essentially findings that he did not actually demonstrate. And yet it was the claim that he had gene mutation that in fact was used to build the linear no threshold dose response model. If you didn't make that claim for gene mutation, you couldn't have built that model. So what we actually have here is a variety of things. We have key findings. We have Muller's gene mutation claim, I believe, has now been proven incorrect. The linear single-hit model was based on Muller's incorrect interpretation. I believe that Muller was deliberately deceptive in his Nobel Prize lecture. I believe that Muller and Stern misrepresented the Manhattan Project findings to promote the LNT. The Bear 1 panel, 1956, in my opinion, committed scientific misconduct in their science paper by incorrectly and deliberately reporting the incorrect data that ultimately to show that they did not want to show the variability and disagreement amongst themselves, and they misrepresented the number of people who submitted reports and the degree of variability that was left in those ultimately to support their interpretation. The Bear 1 report in 1972, I don't find any dishonesty there, but I find error. There may have been dishonesty in the part of the Russells. I don't know that for sure. That's what Selby thinks. But basically, when you correct for that error, it discredits the LNT interpretation, as I just mentioned. So why has LNT succeeded? And why is it still the prominent paradigm? Producing gene mutations by Muller truly was a major advance, and people just flocked to it. So the development just overwhelmed the field. And contemporary criticism was very strong, but Stadler couldn't deliver him the knockout blow. And Muller had much more personality than Stadler, and he was dominating the show. The Manhattan Project was a massive project. It had all the government money behind it, and the support was going to go behind the Stern papers, and it did. The dropping of the bomb frightened the world and made people afraid of any level of radiation. The Nobel Prize that Muller got for reasons that I believe he didn't deserve created a major platform for Muller and his ideology. And Muller used that ideology to promote his causes. And then the Cold War was pretty evident if you happened to live during it, in my childhood anyways, and it was a scary time. The Rockefeller Foundation was very powerful, and they created a separate genetics panel. Now this is really significant. If the genetics panel, you got LNT only because of the genetics panel, because they were hand-selected, because they supported LNT. And you know that if you send your paper to the right reviewers, you're going to probably get it published. If you send it to people who oppose it, you probably won't. It's just kind of how it works in life. And the Rockefeller Foundation handpicked the people that they had funded with their own money and were still funding, and actually, if you read the transcripts, they were bribed during the course of this to get results that the Rockefeller Foundation wanted, with even more grant money, and my papers document this. The NAS basically was king. It still is king today. And basically, you know, when you have prominent people with Nobel Prizes and they have ideological biases and they practice essentially lies and deception, it goes a long way. People are hard to challenge them. So that, in my opinion, the scientific community got the LNT question wrong. Scientific errors, ideological science, and deliberate deceptions led the way, and by really prominent people. Self-interest, and I have letters between members on that committee indicating that they wouldn't mind stretching a point if it meant getting grant money. And I am really written by people on that committee. I've written this and published it in the literature. It really does not speak well of them. And I believe all the errors and deceptions and mistakes were given a pass. The scientific and regulatory communities existing still today, they've failed in their oversight and review and leadership. And so basically, my conclusion is that the LNT for cancer risk assessment was really based on an initial wrong interpretation by Muller. The single-hit model was faulty, and Muller did not deserve his Nobel Prize. The LNT became established based on scientific misconduct by leading radiation geneticists of the Manhattan Project and the U.S. National Academy of Sciences. As you can imagine, I've had some battles with 2 presidents of the National Academy of Sciences. It hasn't been a pleasant relationship, you might say. They defend Muller, they defend the organization that they're president of, and I pretty much lay out the facts, and we duke it out. And so I know that it's hitting home when the president of the National Academy has to go out and take me on. You know that they think it's kind of serious. So I believe ideology and unethical actions of leading radiation geneticists prevailed, and not the science. So that's my story, and I'm sticking to it. And but I believe that this impacts many aspects of our life and decisions that we make, and the risk communication messages and political posturing by people running for office, and what the media purports. I'm not saying that LNT doesn't exist. I'm saying that the people who the whole basis for it, what has been used to do it, doesn't have a factual foundation. Yes. Okay. Well, I'm going to call my presentation over right now, and so I'd like to thank you all very much. And I've got anybody who wants copies of my stuff, I'd be happy to send you PDFs of them, and then we can go forward now.